right, so we'll get started right now, and whoever joins in, I'll let them in. But what we're going to talk about today is just action potentials, and then kind of bring everything together next Thursday. All right. Now, next Thursday is like next week will be our second lecture exam. So, it'll be released after our class. So then hopefully people have a chance to kind of review everything. With this action potentials, there's a couple of things that we got to have couple of ground rules that we need to understand before we discuss action potentials in general. All right, one of the big ground rules is that there are cells that are at rest polarized, meaning that at rest there's a charge difference between the inner aspect of the plasma membrane and the immediate outer aspect outside the plasma membrane. So we're not talking about there's a big charge difference throughout the width of the actual skeletal muscle cell. If I would put this electrode and put it right in the middle right here, there wouldn't be a noticeable charge difference. It is only on the actual edges of the cells where the plasma membrane is found that you will see a little bit of a charge difference. Now, normally, depending on the type of cells, we will have either a -85, -70, or -90, all right, in terms of the actual resting membrane potential, or RMP. These are words that I want you to get to know, because they kind of, if you've ever read an article, a journal article about, in the sciences, and you have a scientific paper, scientists love using acronyms. They'll use acronyms for everything, right? So just keep that in mind, that have to kind of understand these acronyms and what these stand for. So resting membrane potential, what is it? The resting membrane potential is the actual charge difference between the inner aspect immediately on the inside of the plasma membrane and the outer. So meaning that it's about minus 90 millivolts more negative right here when compared to the outside, right? Now, why do we have this RMP, and what does it allow us to do? Well, the RMP right here is caused by a couple little things right here. One, sodium-potassium pump, right? The second thing we're going to see is that there are a lot of leakage channels. Now, the leakage channels that we're going to see are not gated, meaning they're always continuously open. And what we're going to see is that there's a lot more potassium leakage channels in our skeletal muscles and in our neurons, a lot more potassium leakage channels than sodium leakage channels. Now, why is this important? Simple, right? Normally, there's a lot of potassium on the inside of the cell, very little potassium that's in purple right here on the outside. So if you count, you can see that there's a huge difference in the numbers of these purple dots that's supposed to represent potassium, inside versus outside. Now, the reverse is also true for sodium. There's a lot more on the outside than on the inside. Now we have concentration gradient for both sodium, high on the outside, low on the inside, and potassium, high on the inside, low on the outside. Now, what causes this? Well, a lot of it's caused by our sodium-potassium pump. If you look at a sodium-potassium pump, for every 3 sodiums inside, we're going to move it outside. And then in the process, take potassium from the outside and move it in. 3 sodiums from the inside moved out, 2 potassiums from the outside moved in. So the sodium-potassium pump goes against a concentration gradient in both examples here, right? And what else? Well, if I keep pumping sodium out, it makes sense. I'm going to have a lot of sodium on the outside, like we see right here, right? If I keep pumping potassium in, it makes sense that I'm going to have a lot of potassium on the inside, like we see here, right? Now, what happens with these leakage channels? Well, these leakage channels, if you take a look, there's a lot more potassium ones. So you have not only the sodium potassium pump, pumping out 3 sodiums, 3 positive charges, only bringing in 2 potassiums, 2 positive charges. So with this pump by itself, you pump out an extra positive charge, right? By pumping out an extra positive charge, the inner lining of the plasma membrane right here becomes slightly more negative. Because remember, when you lose a positive charge, you don't become neutral, you become the opposite, negative. So that helps explain some of the actual, reasons why it's slightly negative on the inside of our plasma membrane here, because we keep pumping an extra positive charge out. Not only that, what we're going to see is we have a lot more potassium leakage channels. So even though we're pumping some of the potassium in and we're having some positive charge come in, because we have a lot of these potassium leakage channels that are always open, the potassium that we bring in has a tendency to want to go out because there's a lot more of these channels. And it goes out because of the concentration gradient, high on the outside, low on the inside. Potassium wants to leak, and potassium's positive charge. So as it leaks out, you become even more negative. So all of these kind of allows us to be slightly more negative, minus 75, minus 85, when we compare the inside of the plasma membrane of skeletal muscle cells. And this is going to be true with, neurons as well. Slightly more negative in the inside of the cell, the plasma membrane, versus the outside. All right. Now that's at rest. We're at minus 85 millivolts. So that's an off switch, right? So the off switch, when you're resting, everything's chilled. Guess what? The off switch means that it's slightly negative on the inside, minus 85 millivolts. Now, we need this resting membrane potential because it's our way to turn on the muscles, turn on a neuron. So then once we want to activate it, we have a mechanism to turn it on and then turn it back off. So at rest is at minus 85. When we want to turn it on, I'll make it positive. When I want to turn it back off, I'm going to make it negative again. So this is our organic mechanism. So then we can have a power switch for our cells. Now, in order to understand how this system works, we need to kind of go back and discuss a couple of these things that we discussed much earlier in the semester, right? So we have these channels. Some of them are always open, like the one we just saw in the previous slide. They're called leakage channels. There's no gate. They're always open. There's always a little bit of movement of sodium through its sodium leakage channels, and a little bit of leakage of potassium through its leakage channels. So those are one type of channels that we're going to see here in the skeletal muscle cell. The second type is going to be the ligand gated, and the third type, voltage gated. Now, a ligand is a very, very, very generic name for, it can be a neurotransmitter, it can be a hormone, it can be, right? Usually even calcium, right, can be a ligand. So ligands is a very broad category of molecules that can bind to a receptor site and cause some kind of activation. It can be a hormone or neurotransmitter. Those are usually the two main ones. Now, in order for a ligand-gated channel to work, you're going to have the ligand. The ligand, again, right? Hormone, neurotransmitter. You need to have a receptor on the target cells that can bind to the ligand. So if we have a ligand-gated channel, we need to have that gated channel that has a binding site for my specific ligand. And when my ligand is released, only then do you open the gates. So our first example of a ligand-gated channel are what we call acetylcholine receptors. Acetylcholine, I'm just going to highlight this part right here as well. Acetylcholine is usually, we usually use an acronym ACH, right? Now, acetylcholine is a neurotransmitter. We're going to see acetylcholine receptors on the sarcolemma of our skeletal muscle cells. Acetylcholine is the main neurotransmitter at the muscular system of the muscular system. They're the main neurotransmitter at what we call the neuromuscular junction. That's where the nerve makes contact with the muscle. Now, with all of these gated ion channels, ligand gated, voltage gated, right? All of them are going to be closed until some kind of event occurs, which will then open them. For ligand gated ion channels, they're always closed until you have that ligand released, bind to its gate, and then cause that gate to open up. Now, what we're going to talk about today is acetylcholine, right? So neuron is going to release acetylcholine. When acetylcholine, the ligand is released, is going to bind to its acetylcholine receptor. That combination of binding to the receptor causes an activation of the gate, and the gate opens up. Now, the gate is a sodium channel right here. So what we're going to see is the ligand-gated channels are sodium channels, and they only open and close when there's a ligand bound to it. So just keep that in mind. These ligand-gated channels, they're usually the starting point of how we will have a muscle contraction. So just keep that in mind right now, what it is, right? We'll go into a little bit more detail specifically about acetylcholine receptors in a second. The second type of channel that we're going to, well, actually the third type besides the leakage one, is a voltage gated channel. Voltage gated channels are going to be very similar to ligand, except they open and close in response to change in voltage. Meaning when the membrane potential is at -85, there's no change in the voltage. These channels don't open. They only open once you hit a set point. So there's two main types. The first type are voltage gated sodium channels. And then the second type, voltage gated potassium channels. Now in reality, we'll talk about other types, but these are two main players. And then when we start talking about cardiac muscle next semester, that's when you guys will start kind of really discussing calcium. Because next semester we start with the heart and the blood right away. And if you understand this, you're in amazing shape. I think you would actually understand it. So I think you'd be in good shape. So we have voltage gated types of ion channels, voltage gated sodium, voltage gated potassium. Now they only open at what we call a threshold potential. That's that, threshold you have to reach, right? In order to open up these channels, especially for voltage gated sodium channels, in order to open it up, you need to hit -55 millivolts. If you don't hit -55 millivolts, then the channels don't open. So hitting the -55 is incredibly important. Now, the second type of channel right here, voltage gated potassium channels. These channels are going to be open and closed also in response to changes, but it needs to have a slightly different voltage threshold to be reached for it to open. It starts opening at 0 millivolts. And we're going to see one of these causes the nerve impulse or action potential. The second one over here causes the repolarization, so then we can recover. We don't want our skeletal muscles to continuously contract, and we are not able to then, understand it, right? So when we talked about these ion channels, hey, Elias, so we just started talking about ion channels and how they're important for our action potentials and for action potentials in neurons, in skeletal muscles, in cardiac muscles. Now, the important thing to understand is in either one of those cells, skeletal muscles, cardiac muscles, neurons, in either one, the inner aspect of the plasma membrane is slightly negative compared to the outside. Now that's due to the sodium potassium pump. And then the amount of potassium leakage channels, right? That causes us to be slightly negative, minus 85, minus 90, minus 70 in some cells, all right? But it's slightly negative compared to the outside, all right? When we just kind of talked about these two types of ion channels, ligand ones open and close in response to a specific chemical we call a ligand. Ligands can be neurotransmitters, it can be hormones. Now, we will talk specifically a lot this chapter on acetylcholine. Acetylcholine is a neurotransmitter. It's going to be released from a neuron, then it's going to bind with binding sites on the skeletal muscle cells. And once it does, acetylcholine binds to its receptor on the skeletal muscle cells. That receptor is a sodium channel, and it only opens when acetylcholine is bound to it. Then the second type of gated channels that we'll talk about are voltage gated. There's two types: voltage gated sodium, voltage gated potassium. And again, both are always closed until the voltage changes. Remember, we start off at minus 85, minus 90. So in order to open the voltage gated channels, specifically the sodium ones, we need to go from minus 90 to minus 55, right? So we need like a 35 millivolt voltage change, because at rest, the skeletal muscle is slightly negative at minus 90 millivolts. Well, in order to open up those voltage gated sodium channel, we somehow have to go from minus 90 to minus 55. That's a 35 millivolt difference, right? Before we can even open up the voltage gated sodium channels. Now, voltage gated sodium channels will cause us to have depolarization. It will start our action potential. The voltage gated potassium channels are going to be the ones that causes us to recover. So then we can get ready for the next contraction. So let's talk about this, depolarization. And you might see this graph on your next test, something like this graph, right? So what do we see on this graph, and what do we mean by depolarization? You guys know how polarized everybody is on their political views, right? It's kind of disgusting how we can't even talk to each other about anything before somebody gets upset about something we say, right? So when we start talking about kind of polarization and what it means, polarization, whether we're talking about, society, whether we're talking about government, whether we're talking about biochemistry or physiology, polarization means differences. So when we say we have a polarized cells, that means our cells have a charge difference. So we kind of mentioned it, minus 85, minus 90 in some books. Here, because we have the minus 85, we'll just go with this. So in a normal resting cell, resting skeletal muscle cell, it has been turned off, you've been asleep the whole night, so you don't need to move your muscles. What happened? We're at minus 85 millivolts. That's at rest. Now, in order for us to activate the skeletal muscle, we need to have depolarization first. Now, the big thing is this, and I want you to kind of, imagine, how is this happening? We kind of talked yesterday about how we have a muscle contraction, right? How we have, in terms of muscle contractions, how, myosin and actin, right? How these things kind of work together, so then we can actually have a muscle contraction. Well, what I want us to understand is, even before everything, in order for the muscles and the muscle mechanism to shorten and contract, in order for that to happen, you need to be able to turn it on electrically first. So an action potential going through the plasma membrane of the muscle cell, that causes calcium release, and then the muscle contraction can occur. And that's a big thing, is in order to actually allow what I call the mechanical contraction, the actual physical shortening of the cell, right? In order for the cell to physically shorten, you need at first to turn it on electrically. You need an action potential, right? That initial action potential that spreads from one region to the next. That initial action potential is what causes us to eventually have the mechanical shortening that causes the muscles to contract. So this whole thing right here that we're going to talk about next with action potentials, that is what's going to cause something very important to happen. And I'll make it into almost like a two parts, right? This is the first part. Action potentials occur first. Then you have a second part where the actual muscles shorten mechanically. And that muscle shortening is due to two specific proteins in our cells. And what we're going to see is in order for that muscle to shorten, you need this first part to occur. Now, there is a link that allows you to connect what happens to the first part and what triggers the second part to happen. The link is calcium. Now, later on, we're going to somehow put it all together, right? But first, let's talk about these phases of depolarization. Again, in order for muscles to contract and shorten, you need to turn it on electrically by way of an action potential. And then you can have the second part occur, which is the actual physical shortening of the cell. So let's talk about the action potential, the electrical activation first, right? We are at minus 85 millivolts. That's our resting membrane potential at rest, right? Don't know why I've repeated the rest, right? So what happens? What happens is something causes a little bit of sodium to rush in, right? So this initial start right here, right? That is what we're going to talk about coming up, right? This initial depolarization, and I'm going to write this down, right? This initial upswing is due to acetylcholine receptors opening and sodium entering the cells through these ACH receptors. I'm just going to put an ACHR. Right. So again, right now, this initial depolarization right here, that's this. And I'm going to write this down as initial depolarization. Oh, crap. Well, mess that up. Oh, undo it. Perfect. All right. This initial depolarization right here, that initial depolarization, this kind of upswing from rest towards threshold, that's due to what we call our acetylcholine receptors, right? So just keep that in mind, that we have these acetylcholine receptors, and these acetylcholine receptors are going to be closed unless acetylcholine is going to be released, right? So once acetylcholine is released from our neuron, acetylcholine binds to its receptor. Acetylcholine receptors are sodium channels. There's a lot of sodium outside the cell, very little on the inside. Why? Sodium-potassium pump. Sodium-potassium pump allows us to pump 3 sodiums out, right? Out from inside the cell, out into the extracellular fluid space. If I keep pumping all these sodium out of the channel here, it makes sense I have very little sodium inside the cell, very high at concentrations outside. Now the numbers are pretty different, right? Inside the cell, sodium concentration is about 12 milliequivalents. Outside the cell, 144 milliequivalents, 12 times higher concentration outside the cell. Remember, all right, diffusion goes from high concentration of something and then towards lower concentration, right? So here, sodium is very high outside the cell because of the sodium-potassium pump. Sodium then wants to go into the cell because of the law of diffusion. This initial rise from minus 85 towards threshold, it's due to acetylcholine being released. Then it binds to its receptor found on the plasma membrane of our skeletal muscle cell. Once acetylcholine binds to its receptor, perfect. Now we can open up the acetylcholine receptor. The acetylcholine receptor is a sodium channel. So as it binds and opens, you open up a sodium channel. Sodium rushes in. Now, sodium is positive charge. So as sodium goes into the cell, makes sense, you make that area slightly more positive, right? Because you're adding positive charge to minus 85, right? You're starting to make it more positive. Now, what happens next, right? Next, we have, right, the reaching of threshold, right? At minus 55 millivolts, enough sodium, right? Enough sodium has entered the cell through these acetylcholine receptors. Now we have hit the threshold at minus 55. When that happens, voltage gated, sodium channels open up. So right? So when this happens right here, we hit minus 55. Well, this initial depolarization is due to acetylcholine being released from the neuron, binding to binding sites found on acetylcholine receptors found on the sarcolemma of the skeletal muscle cell, right? So now it binds to it, it opens up the channel, and acetylcholine receptors are sodium channels, all right? So once you open up the receptor, sodium comes in due to the law of diffusion. As sodium enters, it makes the cell slightly more positive. Now, if enough sodium enters through the acetylcholine receptors, then you hit threshold. We go from minus 85 to minus 55, right? What happens at minus 55? At minus 55 right here, we hit the threshold voltage and voltage gated sodium channels open. Right? We have a lot more voltage gated sodium channels. So when they all open up, we still follow the law of diffusion. Sodium all rushes in, right? And as it rushes in, it gives us this kind of pinkish depolarization phase, right? So they open up, the voltage gated sodium channels open up at minus 55. Then, right, a lot of sodium rushes in and we go from minus 55 to positive 20. That's the turning on of the cell now. We have flipped that switch. The cell is going to turn on electrically now, right? As it goes, you've turned on the cell, and now we're going to see that we have activated an actual region of our skeletal muscle cell. Now, when we activate one part of the skeletal muscle cell, we're going to see you don't just automatically spread it and propagate it like it's a moving charge, right? What we're going to see is we will have propagation, but, right, it's easier to show you on the next slide, right? The propagation requires one segment, one part of the skeletal muscle plasma membrane to depolarize first, causes the next part to depolarize, which causes the next part to depolarize. You have to go from one to the next, to the next, to the next, right? Now, when one segment of the plasma membrane depolarizes, it's going to carry that sodium that just rushes in to the next segment, causing the next segment to hit threshold and depolarize. Right? As the next segment hits depolarization, the previous segment has their voltage-gated potassium channels open up. Right? As voltage-gated potassium channels open up at positive zero, at zero, right? At that point, yeah, there's a slow activation of these voltage-gated potassium channels. And even then, there's a lot more sodium coming in. That's why it continues to spike. At positive 20, voltage-gated sodium channels close. No more sodium rushes in. Now, voltage-gated potassium channels fully open, all right? Potassium leaves the cell. As potassium leaves the cell, you lose more positive charge. What happens if you lose a lot of positive charge? You become the opposite. You become more negative, right? And you go back to our normal resting membrane potential, right? Again, this occurs after you've already had one initial part of a muscle cell depolarize. So all the sodium's rushing in there. You've activated that one segment. Well, the sodium, it doesn't just stay still, right? In a second, well, let me see if I have that slide right here. All right? So what happens, right? Sodium comes into this segment right here through voltage-gated sodium channels, right? As sodium enters this area, it causes this area to depolarize and it becomes positive. Why? Because the sodium rushes in, right? Now what else happens? As the sodium rushes in this segment, it causes this segment to activate, right? Then the sodium doesn't stay still. The sodium then rushes to the next segment. As it goes to the next segment, internally, it causes this next segment right here to hit threshold. As this segment hits threshold, adjacent to this one right here, because sodium that rushes in moves in this direction. As this segment hits threshold, what happens? Voltage-gated sodium channel in the next segment open up. Sodium in the next segment rushes in, depolarizing this segment. The sodium that rushes in is going to go to the front, anterograde, and it's going to go to the back. It can't tell directions, right? It's like some guys, right? They're going to drive. Now, I don't know how some people get really lost still with all the GPS we have, but you know people get lost all the time. So the sodium rushes in. As it rushes in, it doesn't know which way to go. It's going to go in both directions, right? But what we're going to see is when it goes into one direction, anterograde, the sodium that rushes in here is going to hit a region of the plasma membrane where the voltage-gated sodium channels hadn't activated yet. So it doesn't have to be in recovery yet, right? It is what we call naive. So as sodium goes in this one direction right here, sodium causes the voltage-gated sodium channels to open up because as sodium comes in, sodium's positive charge, right? This area right here isn't activated yet. It's at minus 85 millivolts at rest. But as sodium rushes in from the previous segment, the sodium turns the segment on. Perfect. But it also does something else. It goes to the front, and now we're adding positive charge, right? What happens when you add positive charge? Well, this region right here was at rest, minus 85. Now, all that sodium that came in goes to that minus 85. Well, you're adding positive charge to it, right? Sodium comes into the next segment, it goes to minus 70. More sodium rushes in, goes to minus 55. Causes this segment to hit threshold now, right? As it hits threshold, voltage-gated sodium channels open in the next segment. And now I have moved that action potential from this initial region. Sodium then goes to the next region, causes this next region to hit threshold. Sodium goes in, now depolarizes next region. Sodium that comes in, that causes the depolarization here, moves to the next segment, causing the next segment to hit threshold. As it hits threshold, voltage-gated sodium channels open in the next segment, and now sodium rushes into the next segment. Each segment causes then the next segment to hit threshold, which then causes it to have voltage-gated sodium channels open, and then it causes the next segment to depolarize. It has to go in sequence. Now, I did mention that sodium doesn't know which direction to go, right? It can go to the left, it can go to the right. The thing is, when sodium goes to the right, over here, it encounters a region of the plasma membrane that hasn't activated yet. Their voltage-gated sodium channels didn't open up yet. It's naive, right? It's fresh. So when sodium goes into this one direction right here, it can cause this segment to hit threshold and then cause it to activate. When sodium goes in reverse, though, what happens? When sodium goes in reverse, it hits the region that just activated before. So when it just activated before, we're going to see that it takes a little bit of time for the voltage-gated sodium channels to recover after they've activated. It takes a little bit of time for voltage-gated potassium channels to close and then recover. So when the sodium rushes in, right, in retrograde to the left here, well, it encounters an actual membrane that can't be activated. It's in what we call refraction, right? Same thing here. Sodium rushes in here. It goes to the left. As it goes retrograde, it encounters a segment of the plasma membrane that just underwent an action potential. So the voltage-gated channels haven't fully recovered yet. So even though sodium can go in reverse, when sodium goes in reverse right here, it doesn't stimulate and doesn't cause the previous segment of the plasma membrane to hit another threshold voltage. In other words, then, action potentials can only spread in one direction. It can't go in reverse because every time the sodium goes in reverse, it encounters a region of the plasma membrane that's still repolarizing and recovering. Now, repolarization is due to voltage-gated potassium channels opening up, potassium leaving. So when, right, you depolarize, that's when you have the sodium rushing in right here, right? Now, as the sodium moves to the next segment and causes the next segment to hit threshold, well, this previous segment now is in the repolarization phase over here, right? And since it's in the repolarization phase, voltage-gated sodium channels just open. It can't be reopened again. Voltage-gated potassium channels are still open, right? So you're still having potassium leave. It can't be ready for another action potential yet. So what happens when sodium goes in reverse? It encounters the membrane, the plasma membrane region that is now in repolarization or recovery, right? That when the actual segment here is in recovery or repolarization, you can't re-stimulate it. This allows an action potential to go in sequence from one region to the next, to the next, to the next, but it has to go in sequence. See that? You can't just go from here all the way to there and skip all the previous regions in between, right? You have to go from segment to segment, to segment, to the next segment, and it moves in one direction. Does that kind of make sense now? Why we always say action potentials always moves in one direction, right? Is because, again, right, sodium doesn't have eyes and ears. They can't tell what's right and left, right? Doesn't know what is forward and back. It goes in both directions. But when sodium moves towards the back, when it takes a left turn instead of a right, well, the sodium encounters a plasma membrane that had just activated that's in repolarization. And those voltage-gated channels cannot re-stimulate yet. Now, at the neuromuscular junction, what happens? Right? Remember, for muscles, all muscles require a motor neuron for it to activate. Without the motor neuron being active, the muscle fiber can be huge, it can be incredibly strong, but it can't activate without the motor neuron, right? Now, in the motor neuron, motor means it's going to cause some kind of motor activity, right? So when it discharges, it's a neurotransmitter, which is acetylcholine here. When it discharges it and releases it into the synapse right here, right, the acetylcholine is going to bind onto acetylcholine receptors found on the plasma membrane here, or sarcolemma. Now, keep this in mind, right? There's a region right here we call the motor end plate. I'm going to highlight it and underline it. You might see it on the test, right? Now, I say that and people still aren't going to study it and know it, right? Now, the motor end plate is part of the plasma membrane that is incredibly folded. Why is the folding important? The folding is important because it increases the surface area for the plasma membrane. The more surface area, the more receptors. The more acetylcholine receptors in this region, the easier it is to activate this region. The easier it is for acetylcholine to bind to some of these receptors, right? And then cause us to hit threshold. And then we have voltage gated sodium channels that open up. So keep that in mind, that all of this system right here with the motor end plates is to increase the probability of having an action potential in the muscles, right? Now, whenever we think about skeletal muscles, I want you to remember, I want us to think about our physiology as it was before we had all these amazing weapons, before we had all the restaurants that we can go to, right? In other words, as we were evolving, right? As we were, hunters, settlers, right? And what happens is back then, if you see an animal, if you see even a deer and you don't have a spear with you or anything, well, it could be kind of dangerous. Definitely if you see like a bear, if you see a mountain lion, things like that, right? We are not equipped to fight those animals hand to hand, right? So you're better off being able to flee from them. So we need to be able to stimulate our muscles as fast as humanly possible. And then we need those muscles to stay activated so then hopefully we can get away. So just keep that in mind in terms of why we want to almost have a hairpin trigger so then we can stimulate the muscle easily, right? Well, one of the ways we can make it more likely to have an action potential and then activate the muscle is by increasing the number of acetylcholine receptors. But there's only so much room here, right? So what do we do then, right? Instead of having just an unfolded sarcolemma, where it's a straight line, what we're gonna do is fold it up. Folding it up increases the surface area of this region without increasing the volume, right? Now that I have increased surface area right here, guess what? I have more room to have more acetylcholine receptors. So if I have more receptors, they can, the last thing we want to do is release acetylcholine and not have enough receptors, right? If that happens, it means that some of the acetylcholines have nothing to bind to. It makes it less likely to have action potentials. By having more acetylcholine receptors, we increase the chance that all of the acetylcholine that's released will bind to its receptor. Then we'll have a greater number of acetylcholine receptors open up, a lot more sodium rushing in those receptors, and now I'm likely to hit threshold. If I hit threshold, then voltage-gated sodium channels are going to open up, right? So this motor end plate is incredibly important because it allows us to increase the number of receptors, which will then allow us to increase the probability of hitting threshold and then having an action potential. Again, this is intense stuff. Now, all right, sarin gas, right? If you ever watch 24 or some of the videos about on TV about the sarin gas attacks and some of the organic pesticides are acetylcholinesterase inhibitors. Acetylcholine is a neurotransmitter that's released from the motor neuron, and then it binds to receptors found on the motor end plate of our skeletal muscle cell. Again, I'm going to use these words a little bit more so then you guys get used to it, right? So acetylcholine is released onto the synapse. It's released from a motor neuron. It binds onto the acetylcholine receptors found on the motor end plate. Now, if I want to stimulate the muscle, I'm going to release a lot of acetylcholine. How do I then get you to relax? Well, in order to relax, right here, I need to degrade the acetylcholine. So once acetylcholine is released, it binds to acetylcholine receptors, which are ligand-gated sodium channels, right? Acetylcholine is a ligand. It binds to its receptor at their binding sites. Once bound, it activates and opens up the sodium channel. Sodium rushes in. Enough sodium rushes in, you hit threshold. And then they don't show this right here, but once you hit threshold, voltage-gated sodium channels open, and that's what causes the action potential. But they don't show it. They got lazy. So just keep that in mind, though, that once you hit threshold, you should have a second group of receptors that open up. That's a voltage-gated ones. Now, once you have an action potential that's kind of moved, what happens if we don't want any more contractions, right? What happens then is the acetylcholine that we release binds to an enzyme receptor called acetylcholinesterase. Acetylcholinesterase breaks apart acetylcholine into two of its substituent, starting materials, right? The two substituent starting material is the acetic acid and the choline. And what do? We break it apart and now separate it. It can't do its job, right? What have we done? We've gotten rid of acetylcholine from the synapse. So then it's not just sitting here perpetually activating our acetylcholine receptors, right? Now, what would happen if I destroyed the acetylcholine esterase? Not even destroyed, if I kind of modify it so then it can't do its job. Well, what happened is the motor neurons release acetylcholine and it stays here. As acetylcholine then binds to its site, it falls off, and instead of being broken apart, the acetylcholine will then bind to the next site, causing the next site to open up. And then once released, it goes to the next ligand-gated channel, causing that channel to open up. And then go back to the other one. In other words, it keeps activating your acetylcholine receptors. So as long as acetylcholine is found in the cleft, in the synapse, right? As long as it's found there, it will keep activating its channel, causing, right, more sodium to rush in, right? Causing voltage-gated channels to open up. So then you have an action potential. And then as soon as you recover, another action potential. And as soon as you recover, another action potential. In other words, you keep stimulation. You just, as soon as possible, you stimulate the muscle again. And then you stimulate the muscle again. And you keep on stimulating the muscle. Eventually, the muscle collapses because you stimulate it so often, right? And it starts to spasm. Now, with sarin gas inhibits the acetylcholinesterase in a way that I just mentioned. As you keep spasming and spasming, if it was just purely right? Your appendix, like the skeletal muscles, like your biceps, your quadriceps. Yeah, you have spasms there. It's going to hurt like hell, but you're not going to die. What sarin does is it causes your diaphragm to spasm. And as your diaphragm spasms, you can't take deep breaths. And next thing you know, you can't take any breath, right? Remember, as you take a deep breath, the diaphragm has to contract. Contraction means that the diaphragm moves from your thorax down into the abdomen. As it moves into abdomen, your thoracic cavity increases in shape. Your lungs increase in size, which causes a pressure drop inside the lung. As there's a pressure drop inside the lung, air goes into your lung. Perfect, right? Now what happens if you can't relax it? If you can't relax your diaphragm, the diaphragm stays in the abdomen and you can't exhale. If you can't exhale, you can't take your next breath. And that's how people die, is through the process of suffocation, right? Now, in Syria, we saw this a couple of years ago, where the madman Assad kind of released sarin gas on his own people, right? And the people that died were the kids, right? In Iraq, right? We know that in the late 80s, early 90s, right, Saddam Hussein would release lots of sarin gas onto the population, right, north of Iraq, what they call the ethnic Kurds, right? So the Kurds then were really unprepared for this. How can you be prepared for this, right? Well, they would release sarin, killing thousands and thousands. The only treatment for sarin gas is to have atropine. Atropine reverses the function of sarin on acetylcholinesterase. So it blocks the inhibition of acetylcholinesterase. And that's why atropine works so well. But there's no way in the world those villagers in Syria, the villagers in Iraq, in the Kurdish controlled north, no way they'd have atropine. So you're attacking a population that had absolutely no way of defending themselves, right? That's what you call a madman, right? Now, what have we done now? Again, right? What we are seeing here is the electrical activation. Voltage-gated sodium channels open up here. Sodium rushes in. Goes to the next segment, causes this segment to hit threshold. Now sodium comes in. As you hit threshold, voltage-gated sodium channel in the next segment right here, open up, sodium rushes in. Sodium causes the next segment to hit threshold. As this segment hits threshold, the voltage-gated sodium channel hits threshold at -55. They open up. Now sodium from the outside rushes in because of the law of diffusion, right? Then this segment will be the next segment that opens up, right? Then the next one. What have we done? We have moved that action potential down to the right in sequence, right? Each region causing the next one to activate, which causes the next one, which causes the next one. That is what we call an action potential, right? It's not a moving charge that's perpetually moving like we see in an electrical current. That's why most professors don't like using the term nerve impulse anymore, right? Now, what we have then done is cause the first phase right here, excitation. We've already talked about the contraction. The contraction is a physical shortening, and it's caused by the activity of myosin on actin, right? We call this combination the excitation contraction coupling. Now, what links these two, right? What causes excitation to activate contraction? Calcium, right? Remember that we have these transverse tubules that's connected to the plasma membrane. The transverse tubules, right, connected plasma membrane, and as one region of the plasma membrane, right here, depolarizes, it causes the next segment of the plasma membrane to depolarize, which causes the next one, and then the next one. And then there's a transverse tubule that is connected to the plasma membrane. So the plasma membrane then follows the transverse tubules deep into the cell. As an action potential spreads down the transverse tubules are found as a triad in a group of 3, right? So as an action potential goes down the transverse tubule, the other components of our triad, the sarcoplasmic reticulum, the cisterns, right? The cisterns are these dilated, let me see if I can see the picture right here. All right, the cisterns are these dilated sacs right here, right, that are found kind of sandwiching the transverse tubules. So as an action potential spreads through the sarcolemma, the action potential then goes down the transverse tubules as well, because the transverse tubules, they are part of the sarcoplasmic reticulum, they are part of the sarcolemma, right? They're connected to it. So whatever happens to the sarcolemma right here, the plasma membrane, right? If you have an action potential, you're gonna have an action potential that spreads down the transverse tubules. As the action potential spreads down the transverse tubules, you're gonna have these terminal cisterns holding and storing calcium, right? But as an action potential spreads, that's going to be the signal now for calcium to be released from these terminal cisterns. Remember, at rest, what happens? Low calcium. Because there's low calcium, the regulatory proteins that regulates when muscles can contract, the regulatory proteins are tropomyosin and troponin. Well, what moves tropomyosin out of the way is when troponin activates. And troponin activates when there's a lot of calcium. So at rest, very low calcium. If there's very low calcium at rest, what happens? At very low calcium levels, troponin, right, is not going to shift tropomyosin. Tropomyosin blocks the interaction. So let's show the tropomyosin right here. So right here, right? Here's actin. Actin has a binding sites in yellow. So then myosin can reach up and pull, right? Well, here's tropomyosin. Tropomyosin is a gate. It blocks the connection between myosin to actin. By blocking it, you prevent them from interacting with each other. That happens during relaxation. But keep in mind, the myosin head is already cocked and ready to go. It's already powered up. All it has to do is wait for tropomyosin to move out of the way. Once tropomyosin is moved out of the way, myosin can reach up and pull on actin. On this side, myosin is going to reach up, pull actin to the middle. On this side, myosin is going to reach up and pull actin to the middle. What happens? If everything gets pulled to the middle, then the space between the Z disc is going to shorten. That's exactly what happens when we contract the muscle. Now, right, to prevent unwanted contractions, tropomyosin's in the way. Right? Tropomyosin blocks these two from interacting with each other. When we want a contraction, we need to move tropomyosin out of the way. How do we do this? We do it with troponin, right? When there's no calcium in the cytoplasm, troponin, right, is just going to be at rest, doesn't do anything. Tropomyosin is found blocking the binding sites. But when there is activation, when we want the muscle to contract, calcium will be released. Calcium then binds to this red troponin. Troponin is a calcium binding protein. When calcium binds to it, troponin moves tropomyosin out of the way, allowing actin and myosin to interact. Perfect. Now it can stimulate. So calcium binding to troponin causes contraction, causes the actin and myosin to interact, and now I can contract and shorten. What causes calcium to be released? The action potential, right? The action potential that spreads right here. Action potential that spreads down the plasma membrane. That action potential, as it spreads, goes down to transverse tubules. As it goes down to transverse tubules, that action potential, that charge difference, is going to trigger the release of calcium from these terminal cisterns, right? That's why the action potential is so important. That's why the transverse tubules are so important, right? Now let's just imagine this, and sometimes it's easy to imagine like some physiology if something is missing, what we call a null effect, right? What would happen if we don't have transverse tubules? So imagine we crisscross these, it's no longer present. We still have a plasma membrane, but no transverse tubules. What would happen? You would have an action potential that will still spread on the plasma membrane, but the terminal cisterns right here, they're not found on the plasma membrane, right? So even though there's an action potential that's spread on a plasma membrane, you can't carry it. So then you can activate the terminal cisterns. When that happens, you can't release calcium then, and you can't have a contraction. Why? Because the action potential will only occur on the outside of the cell. You're not going to be able to carry it inwards where the terminal cisterns are found, right? So just keep that in mind. If you're ever confused, imagine what happened if we didn't have these transverse tubules. If we didn't have these transverse tubules, yes, you will still have an action potential down the plasma membrane, but you're no longer carrying it down deep into the cells. And because you're no longer carrying it deep in the cells, you're no longer activating the release of calcium from the terminal cisterns, because those are deep in the cells. And because you're no longer releasing calcium, you're no longer going to have a mechanical shortening. In other words, you'll only have the excitation. You wouldn't have the second phase, the contraction occur. Right? Calcium release, like I mentioned, calcium is incredibly important. It links, right? It connects what happens with excitation to what happens with contraction. Now, I think in my in-person, I do have videos about my lectures in person where I kind of draw a connection, an arrow that kind of goes from excitation to calcium release, and an arrow from calcium release to contraction, right? The sole purpose of all the action potentials of this excitation is to release calcium at a specified time. Then the calcium then causes the contraction. So calcium is the reason why we have an excitation. Calcium's a reason why we can contract. It is that important. Right, any questions so far? Kind of crazy, right? Right. Right, so over here, right, you have an action potential that's spread through the plasma membrane, the sarcolemma, then it goes down the transverse tubules, which then will cause calcium to be released from the sarcoplasmic reticulum's holding tank, which is the terminal cisterns. The calcium that's released then goes and binds to troponin. Troponin then moves tropomyosin out of the way, allowing actin and myosin to interact. All right. Sorry. All right. And if you understand that, there's a sequence right here. And this actual thing is not too bad because it tells you a sequence and how things happen. So a lot of people get confused on reading it. Just follow the numbers. 1, what happens? Then it causes 2, the calcium release. Then it causes 3 to cause a binding to opponent, that kind of stuff. All right. So just don't get overwhelmed by looking at these diagrams and the numbers, which in some people, they do get overwhelmed, right? And then this one's really, really, really small print. So what we're going to see is once calcium binds to the opponent, it causes the cross bridging. Cross bridging is the name of the interaction between myosin head as it connects with actin. Then it pulls. As it pulls, myosin's going to pull actin to the middle here on each side. Now, what else happens, right? Well, as soon as you meet and you pull one time, you release ADP, and a fresh new ATP must bind to it, all right? In other words, you need ATP to dislodge. And not only that, this one kind of shows us how this happens in more numbers. The only thing here is this. They don't show the voltage-gated channels. So just keep in mind, all right? Yes, acetylcholine causes the postsynaptic membrane to open, right? But it causes that to open by opening voltage-gated sodium channels. We're not going to see ligand-gated channels here, all right? We're not going to see acetylcholine receptors here or here, right? You only see it at the motor end plate, at the neuromuscular junction. So they got really lazy. For some reason, they didn't want to show the voltage-gated sodium channels. But keep in mind, right, these number 5 and forward, that's caused by voltage-gated sodium channels. Right. So review this. Muscle relaxation requires fresh ATP, right? In other words, ATP is required to contract and is required to relax, right? ATP, so once you have a muscle's head kind of move out, right, you need fresh ATP to bind to it, the myosin head, and then it can dislodge, and then you can use the power of ATP again. So ATP is required for both relaxation and contraction. Now, the weird thing is this. After all that work we just talked about, right? Action potential, right? Going down to transverse tubules, causing calcium release, binding to troponin, moving tropomyosin and shifting it, allowing the myosin's head to move actin. All we have done is get a twitch, right? That's it. All we've done is a twitch. So here is a diagram for a twitch contraction, right? Take a look. This is when we first stimulate and have an action potential, right? Then it takes a little bit of time before we have full contraction strength. Then after full contraction strength, we have relaxation period where then the contraction is going to decrease in power, right? It's not very strong, this twitch contraction. So if all we have with an action potential and calcium release is a twitch, that's it. How do we get a stronger muscle contraction where we can lift 30, 40 pounds on a bicep curl, right? How do we get that nice, strong, smooth muscle contraction, right? Number one, to have nice, strong, smooth muscle, even real strong contractions, we want to activate what we call motor units, all right? A motor unit is one motor neuron, and you can see a motor neuron right here. Sorry, I gotta make it too big, right? It's gonna come down and then it's gonna split a few times. As it does that, right, one motor neuron can go to many individual muscle fibers, right? So take a look at this motor neuron. It goes to 1, 2, 3, and 4 muscle fibers. There's a 5th that comes down right here, right? Probably a 5th and a 6th one, right? In other words, one motor neuron can go to many muscle fibers, right? A motor unit is one single motor neuron, and every single fiber, muscle fiber, it innervates, it controls, right? So in large muscles, one motor neuron can be connected to hundreds or thousands of individual muscle fibers. What happens if you stimulate the motor neuron then? If you stimulate that one motor neuron, you have hundreds of, and maybe even thousands of muscle fibers that all contract at once. Makes it a strong contraction because you have more muscles that you activate, right? It gives us strength over precision. Think about what happens with the quadriceps. When you're jumping, let's say you're playing basketball, right? And you're jumping to get a rebound. Does it matter if you jump 20 inches to get a rebound? I know most of us are not going to be like NBA players and jump 30, 40 inches, right? 20 is pretty high if you think about it. So can you jump 20? Does it matter if you jump 20 inches or 24 inches or 18 inches? As long as you get the rebound, doesn't matter, right? You jump and you don't really need to be precise usually when we're doing our jumping. Now, you don't want to miss it by over jumping or jump so low that you're not going to get it, right? But what we're going to see is, you want to sometimes over jump more than even under jump, right? We want more strength over precision in the quadriceps. How about in some of the muscles of your hands? Let's say you're writing something, right? And you can't really control the muscle as well, like you're writing with your off hand, your non-dominant hand. Well, it's hard because even small movements can make your letters look weird if you can't control it, right? It makes it almost, you can't even read it if you're trying to write with the other hand, right? Well, those fingers, those hand muscles, you require more precision than strength. So we're going to see in the smaller muscles where precision is required, one motor neuron will go to fewer muscle fibers. So what happens? Well, you might activate maybe 10, 15 skeletal muscles. So it's not going to be strong because there's fewer muscles, but it will be a lot more precise, right? You can't have precision if all you're doing is have one motor neuron fire and 1,000 fibers activate. There's very little precision there, right? So the muscles that require strength have one motor unit, one motor neuron that goes to many, many, many more muscle fibers. The areas where you have precision and you want precision, one motor neuron will go to much fewer muscle fibers. Perfect. Right? Now, what else? Right? We activate more motor neurons. Great, right? By activating more motor neurons, we can activate more muscle fibers. Now, what we don't do is activate all of the motor neurons that goes to the muscle fibers, right? And we'll talk about why coming up, right? Now, stimulus strength and motor unit, and that would be done right here, right? Now, I said that when we have a muscle contraction that we just mentioned, one action potential, it only causes a twitch. That's it. How do we have nice, strong, sustained contractions then? We have it by this, right? What we call threshold, superthreshold, subthreshold, right? Now, if we have a action potential and the action potential down the motor neuron is not strong enough to hit minus 55 millivolts. So remember, you need a 30 millivolt stimuli, right? Before you can actually hit threshold. So what happens if it's a 29 millivolt stimuli? You go from minus 85 to minus 56, right? Well, what happens? Well, what happens then is nothing. No action potential on the skeletal muscle, no contraction. If you have just a threshold stimulus, right? You barely, it's just 30 millivolts. Then what happens? It's enough to have one action potential and one twitch contraction. So how do I have more action? How do we get stronger contractions then? Stronger, more smooth contractions. we have this suprathreshold stimulus, right? The motor neuron releases a ton of acetylcholine. When that happens, right, we have a suprathreshold stimulus. Enough acetylcholine is rushed in, right? The stimulus goes from minus 85, or the voltage, or it goes to minus 85 and immediately hits threshold because it's such a strong stimulus. What that does do is this, right? Once you hit threshold, you'll have an action potential, right? You'll have an action potential. All action potentials are the same. That means it goes from minus 55 to positive 20. Even when you have a very high, type of release of acetylcholine, what we call suprathreshold stimulus, right? Each action potential still is the same. You go from minus 55 to positive 20. So all individual action potentials are identical. Once you hit threshold, you have an action potential, right? If you have a 30 millivolt stimulus that barely hits threshold, you will have the same action potential as if you have a 500 millivolt stimulus that's super strong and it allows you to hit threshold immediately, right? Same action potential strength. So how can we tell the strength, right? How can we tell the muscle to contract it? So if you were lifting a 5-pound dumbbell versus you're lifting and attempting to lift a 50-pound dumbbell, right? How can we tell the muscles to work out the difference between those two? Because in a 50-pound dumbbell, you're going to need to, really have effort, right? You need to activate more muscles. You need to have some other thing happen as well, right? And I'll talk about that right here, right? What we're going to see is the stronger the stimulus, the more action potentials that are triggered. That means what? If I have a threshold stimulus, I'll have one action potential, and that action potential is the same no matter what, right? Now, if I have a subthreshold stimulus, nothing happens because you never reach threshold, right? Nothing, no action potential at all. Now, how does the body tell the muscle then to contract and lift something heavier if all action potentials are the same? Well, how it does so is not that one action potential stronger than the next one. No, if we want to contract the muscle and carry heavier weight, what the brain will do is it will cause more action potentials. So the stronger the stimulus strength is, the more action potentials it triggers, right? So what we're going to see is if you have a strong stimulus, right? If you're trying to lift 50-pound dumbbell, you're going to need a stronger stimulus. How does a brain allow us to do this? If each action potential is of equal strength. Simple. Instead of having just one action potential and you're done, the brain sends a signal to have as many action potentials as we can, right? That's what we see here. We're going to lift something stronger. I have one action potential, causes this contraction, right? Then immediately a second action potential. The second one causes a stronger contraction. And then a third action potential, causing even a stronger contraction after that. Perfect, right? Now, there is a maximum response, meaning what? Let's say you can lift a 50-pound dumbbell, right? And now you're going to, there are a lot of people around, so you kind of want to show off. And you start trying to do 60 pounds, and you can't do it. But you think, you can do it because there's so many people around, you want to show off, right? So what happens? You're trying to do the 60-pound dumbbell. So what happens is, right, even though your maximum is 50, right? What happens is you can't go beyond the maximum. So even if I have a lot of action potentials, right, at short time frames, what happens? I have an action potential. Now I have really strong contraction. And we'll start over here, right? So you start off here with the first action potential, boom, right? Stronger contraction. Second action potential in a short time frame, causing an even stronger contraction. And now you have a third action potential in a pretty short time frame, causes an even stronger connection. At maximum, you had the fourth action potential, right? Causing a stronger contraction here. At that point, you're already at your strongest level, right? You've trained, and after training, you have managed to lift 50-pound dumbbells. That's your strongest contraction possible. If you try to go 60, you can't go beyond 50 because you're not strong enough. And that's what we see here, that there's a limit to how strong of a contraction we have, right? And that limit is our maximal stimulus strength. Even if we want to go beyond our maximal stimulus, we can't, because we're limited in the amount of motor neurons we can recruit, and we're limited by the amount of and strength of the muscle fibers within those motor neurons, right? I will stop right here. And then I know this is intense stuff, but you just can probably tell I like it, right? You know, it's a lot, you know, I mean, I enjoy it discussing it because this physiology, it's kind of allows us to think, right? And that's an important thing I want you guys to do, is think things through. And if you understand that process of critical thinking and understand how this works, it starts off that basis that now when you are in your field, whether you're an OT or you're an amazing RN, right? You'll be able to think your ways through it. So then you are ready in case something happens, right? There's my trauma surgeon mentor that always has these amazing sayings, right? And you never understood it until you really understood it. You are, you know, on your third day of trauma rotation, you're exhausted, and then it happens that you understand it, right? And one of his sayings is this: you can't see what the brain doesn't know, right? If you don't understand what is going on in front of somebody, you, even though they might be having a seizure, even though they might not be doing, you know, breathing, even though you might be treating them with something and there's no response. If you don't know it, if you don't know the, you know, what possible things could happen, your eyes can see it, but it really doesn't see it, right? So just understand that. If you didn't know what a seizure was, right? And you see somebody on the street having a seizure, but you don't know it, what do we see? You see just somebody kind of moving around, right? That's it. You don't know that they could be in real deep trouble. So this starts the basis of just our critical thinking. So then you understand that, you know what, you guys can handle this, right? Especially you two. I have a lot of confidence in both of you. Any questions? And by the way, if you heard some like stomach sounds, that was my dog, right? He's either snoring or grumbling or having some loud stomach sounds. So